Welcome to another video. In this video, I'm giving the general overview of the complementary DNA and how we can synthesize the complementary DNA. What is the importance of the complementary DNA and what are the components that are required for the synthesis of the complementary DNA? We will start from the basic that is what do you mean by the cDNA? What is actually cDNA? So cDNA is nothing but just a complementary DNA, and the cDNA is synthesized by using RNA as a template. So basically, here we don't need any type of any type of DNA to prepare the complementary DNA. Here we just need an RNA molecule, and which can be converted into the cDNA, which is complementary to the particular RNA. So the whatever the information present in our RNA molecule will be passed through to the this cDNA. So there are a special type of enzymes which converts this RNA into the complementary DNA, and they are called as like reverse transcriptase. So I will explain in further slides what is reverse transcriptase actually. But right now you just understand like reverse transcriptase is a type of enzyme, like a type of enzyme which converts the RNA, or which reverse transcribes the RNA molecule into the complementary DNA. So this is how the cDNA is synthesized, and this whole process is called as reverse transcription. So the information which is present in RNA, which can be converted into the cDNA by using the reverse transcriptase. This is called as reverse transcription. So basically, the cDNA molecules are very smaller in size as they are reverse transcribed from the RNA molecule, and also this cDNA also lack of don't have any kind of non-coding sequences. Non-coding sequences are absent. I mean introns are absent are as we are isolating the total RNA from the cell, so that the RNA molecules are already processed in the cell, so that they don't have any non-coding sequences. They non-coding sequences here. That's why the cDNA also don't have any non-coding sequences, and that's the difference between the genomic DNA and cDNA. So, what is the importance of the cDNA? Why we are preparing the cDNA? So, as we know, there are different applications are there, but I am here enlisting only the main, which are major ones. So, first is the detection of RNA viruses. So, as we know, RNA is single-stranded molecule, and if you want to detect any kind of RNA virus, first you have to convert that RNA molecule into the cDNA, complementary DNA, and then you can use primers for that complementary DNA to amplify the particular gene or gene of a virus. So this is how you can detect the RNA viruses. So it is impossible to detect the RNA viruses without converting to the cDNA. So that's why it is very important. And the second application of means the cDNA is that, like when you are want to study the real-time expression analysis of the particular gene, so that time you have to convert your RNA molecules into the complementary DNA, and then you can design the primers for your target genes, and then you can check the expression of the particular genes. And in that case also, it is very important to convert RNA molecules into the cDNA. So also in this case, cDNA is very important. And third one is the cloning of eukaryotic genes in the prokaryotes. As we know that the prokaryotes don't have any kind of non-coding sequences in their genes, so that the only the exons are present in prokaryotic genes, and there is no introns are present. So suppose you want to clone the particular gene, eukaryotic gene into the prokaryotes. So first you have to make sure that the gene don't have any introns. So that's why here the best part comes is the cDNA. As cDNA we are preparing from the RNA molecule, hence there will not be any kind of introns present in the cDNA. That's why if you want to clone the particular gene, it is very necessary to convert into the cDNA, and then you can use that gene, mm, for cloning purpose. This is why the cDNA is very important. Now, moving towards what are the different components used for the preparation of cDNA? What are the components that we require for the synthesis of cDNA? So, cDNA synthesis involves the buffer, the DNTP, of course. If you want to elongate any kind of genetic material, you need deoxynucleotide triphosphates, and also you need primers, as we are using the reverse transcriptase, and which requires the primers to as we know that like 3 prime hydroxyl group should be free, so that this reverse transcriptase can prepare the rest of the strand by adding the DNTPs. This is why the primers are important, as this in simple terms that reverse transcriptase cannot start the de novo strand. So in case of the RNA polymerases, as we know in transcription, they can start the transcription without need of any primers. But here, the reverse transcriptase and DNA polymerase always requires the primer, and the primers should also have the free 3 prime hydroxyl group, so that they can extend it by this reverse transcriptase. And also the components of cDNA includes the RT enhancer. It is nothing but just preparation of some DNases enzyme. So if your RNA is contaminated with RNA is contam- contaminated with the DNA. So this RT enhancer will digest the DNA molecule, and your solution will be free of any kind of genomic DNA contamination. That's why RT enhancer it is also important. And of course you need RNA. Without RNA, how can you prepare the CD? And nuclease, I mean nuclease free water.